Here we are with fMRI part 4 of I don't know how many anymore. But here we are, we're talking our way through imaging the brain's physiological response. So we talked about how the scanner takes protons processing and turns them into images, and we talked about how oxygenated versus deoxygenated blood gives you different signal strengths. And now we can look at tying the shifts in signal strength to actual neurocognitive events. And for this, we are going to talk about a pair of goggles, swim goggles, that played an outsized role in the history of functional MRI. So Peter Fox, in this 2012 article in Neuroimage, gives a nice overview, an anecdotal overview of some of the really early work establishing the functional MRI, the possibilities with localizing the brain response. And so here's a picture of Peter Fox himself. So this was, I made a little sketch off of a photo that was included in that paper. And so here Peter Fox himself is lying down with these goggles on. And these are these visual stimulation goggles that were made by a company called Grass Electronics, I believe. And they were literally just swim goggles with some LEDs, a little circuit board on the inside of the eyes with LEDs in there so that you could flicker them and examine the brain's response and do psychophysics type experiments. So photic stimulation, just meaning they had flickering lights inside these goggles with the goal being to examine the brain's occipital lobe, V1, area V1 of the occipital lobe, and show that you can characterize the brain's response. So Fox himself was using it at first, this is him about to be put into a PET scanner, so a positron emission tomography scanner, but we are interested in its use in functional MRI. And so apparently Fox loaned out the goggles. He got basically before it was actually went into production, he wrote to the CEO of the company saying that my residency only lasts so long and I need these goggles, and she sent him sent him a pair and said, here, they're yours. He sent them off to Kwong et al., who published a paper in PNAS in '92 using these goggles. They had people, participants in the scanner with flickering lights. And so here I've drawn my own version of their major figure. So, this is after figure 1 from their paper. Non-invasive real-time MRI mapping of V1 activation during visual stimulation. So since this is my impression of it, I think we can get around nice little copyright issues. So what we're looking at is a block design, we'd call it. So in a block design in MRI, you have the same thing happen to the person for a while, so for a chunk of time. So here are 20-second periods where there was no flickering going on in the goggles, and then here we've got 20-second periods where there was flickering, where the goggles were stimulating the eyes with light. And so here we're looking at what we call this one, an overhead slice of the brain. So the back is the visual cortex, and this here is the baseline image. So any of these other images are looking for a change from baseline. And so this allows them to account for the fact that in this old scanner that they were using, their signal intensity was actually stronger, closer to visual cortex, just because of the way their head coil was constructed. But we can see that relative to this baseline, whenever we've got the goggles flickering, we've got more signal, more oxygenated blood going to the occipital cortex. And so Kwong et al. Were able to characterize and show that you can localize signals to particular brain regions using this technology. Very exciting. The same year, another group using the same goggles, Blamier et al., also 1992. Again, I've made my own impression of my own drawing of their figure that captures all the basics, but you can go download their article if you want to see the real thing. We've got time on the x-axis because what Blamier et al. Did was to show that the signal change, like you didn't need to do a block design. You could get away with an event-related design and still see changes. You didn't need to average over 20 seconds worth of recordings of images in order to detect a change, especially in area V1 visual cortex, where brief flickering in this case, yielded a rather substantial difference from the baseline. I believe this is a % change from the baseline. And so, yeah, we can see that there's a time-limited response that at some point after the flickering, after the visual stimulation, so the stimulation, the visual flickering only lasts 2 seconds, but we can see that the brain response lasts for several extra seconds. And so we're getting close here to just like connecting all the dots from like how we start with a big magnet and end up with some insight into cognitive processes in the brain. So, the pathway that we're taking and this getting from neural activity. Here we've gotten all the way to the blood response, and now we need to make like that final leap to say that, okay, we can interpret this as reflecting neural activity in the underlying tissue. So the goal is that we want to use the scanner to image physiological changes that are correlated with neural activity. So in this case, blood flow. And making action potentials. The work of neurons is hungry work. Neurons need a lot of ATP and glutamate in order to fire and communicate with each other, and so a lot of the energy in the rodent brain, for example, goes towards restoration of ionic concentration gradients after action potentials, restoration after excitatory postsynaptic potentials, inhibitory postsynaptic potentials, and then, even just holding neurons at their resting state takes energy. So the brain's a hungry organ. And making ATP and glutamate means you need glucose and oxygen. Oxygen gets delivered via hemoglobin, and the vasculature can send this oxygenated hemoglobin to where it needs it. There's a very fine-grained set of vasculature that suffuses the brain, that perfuses the brain, and sends the blood where it needs to be. So there are some. So this whole idea is the idea of neurovascular coupling, and it's not a perfect correlation between neural activity, like action potential activity. There is evidence that sometimes you can get changes in blood flow that's not directly related to the immediate metabolic needs of the local neurons. You can have neurotransmitters released locally that alter capillary flow in a region. You can have, so Sorotin and Das had an interesting paper in 2009 showing that you can get blood flow increasing for an anticipated stimulus even when there isn't a concomitant increase in local neural activity. So there's still a lot we don't know about the relationship between neural activity and the vasculature response, but it's an interesting literature. We do know that neural activity in an area will lead to increased blood flow, but there are these other things that can also alter blood flow sometimes. Okay, so one more slide, I think. Yeah, so the hemodynamic response. So this is the final piece of it. So we saw this in sort of choppy form in the Blamier slide a few slides ago, but this is sort of an averaged, a sort of schematic overview of the hemodynamic response of the brain. So when you have neural activity in a particular brain region, it leads to a cascade of processes, including the delivery of oxygenated blood to the area. And there's a slow onset to this process. So what happens first, so let's say, so I have this labeled as cognitive event, because, it could be that lights are flickering and your perception of those lights is the cognitive event. Or we could have some other kind of experiment where just we know that something happened at this moment, that we have some evidence that the brain started doing something that it wasn't doing a few seconds earlier. Like a stimulus comes on the screen and there's got to be some processing that happens. So what happens first is that there's a very short window where you see an initial dip in signal intensity, and that's because the local blood is getting deoxygenated, and so there's a decrease, so that hemoglobin becomes magnetic and starts disrupting with the local signal, and you get this initial dip, but then you get this flood of blood into the area with its rich oxygen, and so for the next you get a peak about 3 to 6 seconds later, depending on which brain region you're in, which then falls back down. You get a post-stimulus undershoot. Could last 12 to 30 seconds, and then eventually a recovery. And that's our whirlwind basic, super basic overview of functional MRI. And so we're going to try and use these pieces, build upon these pieces to talk about some more advanced analysis techniques like multivariate pattern analysis. But that's for next time, and I will see you then.